Imagine having a tiny device that can mimic how real human organs work. That's what Organ on a Chip, OOC, does. These systems are like little labs that can recreate the environment of an organ, helping scientists test drugs and study diseases. They're a big deal because they can improve research accuracy while reducing the need for animal testing. But of course, they're not perfect, so let's explore both the pros and cons of this technology. OOCs reduce the need for animal testing. This is a major win for two reasons. Ethically, fewer animals are harmed in research. Scientifically, we avoid a problem called species variability. Think about it. What works in a mouse doesn't always work in humans. OOCs use human cells, making them far more accurate for predicting outcomes. Secondly, OOCs improve drug testing. By mimicking human biology, these devices give us more reliable data about how drugs will perform. This accuracy saves both time and money during clinical trials. Next, OOCs simulate real-life body conditions. They can recreate how organs interact and respond to things like drugs or diseases. Imagine a mini-human system in the lab. This capability opens doors for more comprehensive studies. Finally, OOCs enable personalized medicine. By using a patient's cells, researchers can test treatments to determine the best option for that individual. This means tailored care and improved outcomes. Now that we've discussed the advantages of organ-on-a-chip technology, it's time to take a closer look at the challenges. Like any cutting-edge innovation, OOCs come with hurdles that engineers and researchers are working to overcome. Let's explore the main ones together. However, there are many disadvantages of organ-on-a-chip technology. Some of those would include technical challenges. Designing an organ-on-a-chip is incredibly complex. Think about it. It requires advanced engineering techniques and highly specialized materials. These factors make the development process slow and often quiet. Next are the biological limitations. While OOCs are impressive, they aren't perfect replicas of real organs. For example, they might miss subtle, yet important interactions between cells. This can sometimes lead to less accurate results compared to studying an actual organ. Finally, let's talk about cost and accessibility. OOCs are expensive to create and maintain. This high price tag limits their availability, especially for smaller labs and institutions. As a result, their use isn't as widespread as it could be. As early as 1907, 2D cell culture was being used to study tissue, and has since been the main design scientists use to study everything biology-related. However, the disadvantages of the medium, such as the lack of a cellular matrix, mean that the two-dimensional design does not accurately mimic the true physiology of a given organ. This led to the 3D cell medium design being developed in the late 20th century. Being a better design to accurately mimic organ physiology, studies using 3D cell medium achieved far better results. There are still flaws with the design, such as being batch-dependent, varying from batch to batch. Isolating proteins and nucleic acids is also much harder in a 3D cell medium. These problems led directly to the invention of the organ-on-a-chip technology. The addition of microfluids to biological structures on a chip effectively created organs on a chip, allowing for the study of human organ functions in different conditions at a large, efficient scale, revolutionizing high-throughput drug screening. From the original 2D cell culture design, we now have technology advanced enough to nearly simulate an entire human body on a single chipboard. A rough timeline of OOC's history is as follows. In 2007, students were able to recreate the function of a human lung on a mechanical chip, which inspired a professor to create an organic lung model on a chip with the students in 2010. Now a company, Emulate, has begun to commercialize the technology in 2014. Now in 2023, OOC technology is being used alongside animal studies in FDA-approved settings. For the manufacturing process of organs on a chip is simple to describe, but can get more complex the more you dive in. It starts out the same as tissue engineering does, whereby you take cells that you want to replicate from the body, such as the various heart or liver cells you might want to test your therapeutic on, and put them through cell expansion to increase the amount of cells that you have without having to take more from the host. Once you've fully grown your target cells, they are then transferred onto a scaffold, which will be different depending on each cell, replicating the extracellular matrix that is usually lays on inside of the body to hold them together. However, unlike tissue engineering, this is where the process stops because with organs on a chip, you will usually have a smaller number of cells. After they are transferred to the scaffold, they are then put onto the chip, which can either be by themselves or in a system to simulate what the therapeutic might target. Like what is represented with the picture organ-on-a-chip technology is the cell culturing microenvironment that mimics human cells' environment using encouraged cells to simulate a human organ or tissue into a microfluidic chip. Different types can be grown to recreate specific functions, mechanics, and physiological responses of that organ. Some use cases of organ-on-a-chip technology are lung on a chip, heart on a chip, kidney on a chip, liver on a chip, nerve on a chip, and skin on a chip. Without a doubt, versatility is one of the advantages organ-on-a-chip devices offer. The versatility to replicate such a wide range of biological mechanical signals, enabling limitless growth proven in the last few years. Importantly, considerable advances have been achieved in the creation and utilization by multiple companies and all-around research. Some of the leading companies include Axosim, TARA Biosystems, Alveolex, Tissues, and Nordis Bio. All use organ-on-a-chip technology, but with different applications and purposes. Organ-on-a-chip is revolutionizing personalized medicine, drug development, and disease modeling while offering ethical alternatives to animal testing and clinical studies. However, organ-on-a-chip will face constant limitations with the difficult challenges of creating complex multi-organ systems. The future of medicine may very well rest on a tiny chip.